Okay, here we go with our second set of video notes in the Chemistry of Life series, this one on carbohydrates and proteins. Protein structure is they're made up mostly of, like I said, amino acids, and they bind covalently to form peptide bonds, and then just like you have mono and disaccharides and so on and so forth, you can have dipeptides and tripeptides, and then polypeptides. So this would be like a big chain of amino acids linked together, and then polypeptides can combine to form a protein. So down here we have a little computer rendering of what a protein might look like, and you can get lots and lots and lots of different shapes of polypeptides. This one happens to be made up of lots of different spirals, so each of these different color spirals is a different polypeptide. So you have this picture in your little handout, you might want to attach that here so you can refer to this, but a polypeptide, a single polypeptide, would be each of these things right here, lots of different polypeptides. And then those different structures, whether it be spiral or sheets or folded sheets, they form a 3D protein, kind of like a globular structure. And the thing about this globular structure is you can undo that structure by stress. So if you heat a protein, that might undo the structure, or if you freeze it, some proteins don't react well to acid, so if you were to lower the pH and put it in an acidic environment, it might undo and break some of these bonds, and that might screw things up. So there's ways you can undo this 3D structure. One system of proteins that really uses this 3D structure are things called enzymes. An enzyme is just a protein that can help speed up a chemical reaction. Basically, the enzyme acts upon a substrate, right? This is what the enzyme acts on, and it fits that specific substrate like a lock and key. So every substrate, everything that's going to get changed, has a very specific enzyme that goes with it. Right? And along with that is every enzyme has its own ideal conditions. So you could think of the enzyme lactase is an enzyme that likes to break down the sugar lactose. So if this were the substrate lactose, the sugar you have in milk, lactase being the enzyme that breaks it down, that lactose combines with the lactase, and that does some biological magic and helps break that lactose into two smaller sugars that are easier for the body to digest. Without that enzyme, that would take a long, long, long time for that to happen, and your body wouldn't be able to use that sugar as well. However, if that enzyme were to lose its special 3D shape, then the key wouldn't fit the lock anymore. We call this being denatured. So if the enzyme were to become denatured, then that lock wouldn't fit the key anymore, and then the enzyme wouldn't work. So for example, again, this is a diagram you have. You probably want to attach it to your notes. You see here you have your enzyme and its substrate, and it fits it. Right? You can see it's a lock and key, it's supposed to fit, but we're applying heat. And that heat is breaking some of those bonds. It's like I went up to your house and I smashed on the lock with a hammer. Well, I smashed up that lock inside. Now your key doesn't fit anymore. And if your key doesn't fit anymore, are you going to be able to get into your house? Obviously not. So no, this substrate leaves unchanged. Unlike in the previous diagram, it broke it up into smaller ones. That is unchanged. So for example, on people that are lactose intolerant, their lactase enzyme doesn't work, so they can't break down that glucose. So if you denature an enzyme, it doesn't work anymore. You do that through stress, heat, temperature, whatever. Every enzyme has its own set of special conditions. So some enzymes can be undone by pH, like increasing acidity. Some can't. Some are made or kind of evolved for being in very acidic environments, like the enzymes that are in your stomach. They're fine with lots of acidity, versus other enzymes might not be.